Hello and welcome back to my YouTube channel. In this video, I'll be discussing vesicular mediated transport in much detail. So let's dive into the lecture. By the help of this vesicular mediated transport, materials are taken up by the cell through membrane mounted vesicles. Okay, so materials are taken up by the cell through membrane mounted vesicles. This vesicular transport mechanism is further divided into the endocytosis and exocytosis. Okay, so let's start our discussion with the endocytosis first. This endocytosis is further divided into the three types: pinocytosis, which occur without the involvement of actin or clathrin molecules. Second is the phagocytosis, which occur with the involvement of actin molecules only. And last is the receptor mediated endocytosis, which occur with the involvement of clathrin molecules. This was just a brief overview of all the types of endocytosis. Let's dig into the pinocytosis first. What is pinocytosis? It is also known as cells drinking process. What happens in the pinocytosis? Indentations are formed in the cell membrane. Okay, so these indentations gets deeper and deeper until detaches from the membrane. Okay, what happens? Indentation is formed in the particular part of a cell membrane, and this indentation gets deeper and deeper until detaches from the membrane. These indentations are filled with the extracellular. These vesicles are filled with the extracellular water along with some solutes. That's why, hence, this phenomena is known as cells drinking process or pinocytosis. So this was all about pinocytosis. Let's move on to phagocytosis. This phagocytosis is shown by phagocytes. Phagocytes include macrophages and neutrophils. Okay, what happens in the phagocytosis? The membrane of WBCs move around the invading particles such as bacteria or any other parasites. Okay, what happens? The membrane of these phagocytes move around the invading particles until the end of this membrane fuses with each other. Okay, and on the opposite side of this fusion, there will be the rearrangement of actin molecules, and these actin molecules pull the part of membrane inward, due to which the vesicle is formed, and this vesicle now gets detaches from the membrane, and this vesicle is known as phagosome. This phagosome then attacked by the lysosome. Okay, so this phagosome is then attacked by the lysosome, and these lysosome release their enzymatic content and causes the digestion of invading particles. So this was all about phagocytosis. Let's talk about receptor mediated endocytosis. This is shown, this occurs within the hepatocytes. What happens? After the digestion of fat, LDL is formed. This LDL is taken up from the blood into the cytoplasm of hepatocyte by the process known as receptor mediated endocytosis. On a particular part of cell membrane of hepatocytes, there is the high concentration of LDL receptors. Okay, so there is the high concentration of LDL receptors. As soon as these LDL receptors bind with LDL molecules or cholesterol molecules, then these receptors sends the signals into the cell, due to which these clathrin molecules, special type of protein known as clathrin molecules, bind with the inner part of the membrane. Okay, what happens? As soon as these LDL receptors bind with LDL molecules, these LDL receptors then sends the signal inward, and after, due to the signals, certain special types of proteins bind with the inner part of the membrane and causes the pinching of this part of membrane, due to which pits are formed. These pits are known as clathrin coated pits. Okay, so these pits are known as clathrin coated pits. So these clathrin keeps on pinching this part of membrane, due to which vesicles are formed. Okay, so vesicles are formed. These are known as clathrin coated vesicles. Okay, so these vesicles are known as clathrin coated vesicles. By the action of some enzymes, these vesicles then gets detaches from the membrane. Okay, So these are the vesicles. You can clearly see in this diagram. Okay, and these vesicles then binds with the another type of vesicles which contain enzymes. Okay, so these vesicles bind with the another type of vesicles which contains enzyme, and these enzymes are involved in the releasing of LDL and cholesterol from this vesicle. Okay, so LDL and cholesterols are released from their vesicles, and receptors, LDL receptors are restored, are then implanted within the membrane. Okay, so this was all about the receptor mediated endocytosis which occurs within the hepatocytes. Okay, so which occurs due to the involvement of clathrin molecules. Okay, now let's talk about the exocytosis. Certain materials such as hormones, such as neurotransmitters, are released by the cell through the process known as exocytosis. Okay, so it helps the release of content from the cell. What happens? Consider, suppose this is the beta cell of pancreas. Upon stimulation, what happens? Certain calcium channels are activated. Okay, certain calcium channels are activated. Due to the activation of these channels, there will be the influx of this calcium within the cell, and within the cell insulin is present. Okay, so there are some vesicles which are filled with insulin. Okay, so which contains insulin, and this calcium binds with these vesicles and causes the fusion of this vesicle with the membrane. Due to the fusion of this vesicle to the membrane, this part of vesicle and membrane dissolves, which causes the release of content from the vesicle, from the cell. Okay, what happens? Suppose this is a beta cell. What happens in these cells? Upon stimulation, certain calcium channels are activated. These calcium channels causes the activation of this calcium channel causes the influx of this calcium into the cell, and we know that within the beta cell vesicles are present. These vesicles are filled with insulin, and this influxed calcium ions bind with these vesicles, causes the fusion of this vesicle with the cell membrane, due to which the insulin is released from the cell. Okay, so this same mechanism occurs in the release of neurotransmitter as well. So this was all about vesicular transport mechanism.